Okay, let's start. So welcome everybody to the monthly webinar of EUROTOX, and thank you for attending. So today we will have a presentation given by Professor Giorgia Del Favero on cell deformation in chemical carcinogenesis. So Giorgia graduated in 2008 in pharmaceutical chemistry and technology at University of Trieste. And then she completed a postdoc at abroad, and she joined the Department of Food Chemistry and Toxicology of the University of Vienna in 2014. She's now having she's an independent group leader at this university, and in 2019 she was appointed head of the core facility multimodal imaging, and in 2022 tenure track professor for toxicology at the Faculty of Chemistry of the University of Vienna, where she is currently active. So our main research interest is the study of molecular mechanisms of toxicity affecting cell morphology and mechanotransduction. And I think also, Giorgia, you are the president of one of the specialty sections of EUROTOX.
Exactly, carcinogenesis.
Exactly. So thank you very much for giving this webinar. And I remember to all the participants that the question will be only in the chat. There will be 45-minute presentation, 15 minutes of question. Put your question on the chat and I will read them. And please be sure to turn off your microphone. Thank you very much. The floor is yours, Giorgia.
Thanks, Marco, for the kind introduction and invitation. And thanks to all the colleagues for taking the time and attending the webinar today. It is a real pleasure to be here together with you. So maybe some of you might have wondered why I decided to focus actually a webinar on toxicology on cell deformation. But actually, changes in cell morphology are way more than simple attributes of cell shape. Over the last year, it has become clear that changes in cell morphology are tightly linked to carcinogenesis. And actually, if you think how a tumor develops, you have a lot of changes of cell shape. Think, for instance, about the tumor mass increase, how this is used, also, for instance, for diagnostic purposes. But you have also a lot of cell deformation if you think about metastasis formation, for instance. Think about how the cells have to squeeze to reach the blood vessels for intra or extravasation. And think about the difficulties and the differences for metastatic cells to colonize a secondary tumor site, for instance, with different extracellular matrix stiffness. I'm thinking here, for instance, about bone tissue in comparison to brain tissues. These are tissues that are really very, very different. Last but not least, we also have the fluid shear stress. Think about what happens in the luminal organ, but also about what happens in the bloodstream. And over the last, I would say, 20 years or so, it is becoming clear that these changes of the mechanical tumor microenvironment are not just changes of cell shape, but are also very important signals. And these are essential to actually support tumor progression or also, for instance, chemoresistance. And please believe me when I tell you that these are just few examples of all the works that have been published in the last years, really describing these very intriguing molecular mechanisms of carcinogenesis. So how is this possible? How can actually cells manage to cope with all these different physical cues in the framework of carcinogenesis? Well, this is because cells are very good at doing something called mechanotransduction, which means transforming physical cues into biochemical signals. Now, with the help with this beautiful illustration that was done from my PhD student, Dennis, I would like to show you and introduce to you some of the main players that are involved in mechanotransduction. So starting from the cell surface, the first mechanosensor that we encounter is probably the cell membrane. Think how cell is dependent on cell membrane composition. For instance, if you think about migration and the formation of filopodia, lamellipodia, how the cell membrane has to be flexible and actually stretch to enable cell movement. But this is not all. The cell membrane is also very important in harboring essential mechanosensitive structures. I'm thinking here, for instance, to the caveolae. Caveolae are actually membranes in vagination that serves as membrane reservoirs and actually stretch whenever the cells needs it in order to make available a bigger surface when the cell has to move and adapt very quickly to external stimuli or in case of movement. And on top of it, we also have mechanogated ion channels. One example are, for instance, the piezo channels that will open in response to cell deformation, letting, for instance, calcium increase in the cell in response to changing cell shape. So obviously, if we move down toward the intracellular compartment, we also have adhesion proteins, like for instance, integrins, that are connected with the tightly meshwork of the cytoskeleton, actin, but also intermediate filaments to the nuclei. And the nucleus, last but not least, I would say. The nucleus is also one of the main cellular mechanosensors, is equipped with a very sensitive nucleus skeleton, and in the nuclei, we also have the activation of transcription, which is potentially regulated by mechanical stimulation, thanks to the activation, for instance, of mechanosensitive transcription factors. So all of these to bring inward and outward mechanical signaling from the cell surface to the nucleus and back. So for today's presentation, I would like to focus exactly on these mechanisms of toxicity. In the first part of the talk, I would like to introduce actually some work we have done about chemical modulation of mechanotransduction. Two different works with different focus. One more focus on the tumor cells and the other one on the sensitivity of the non-transformed cells. The second part of the talk, I would like actually to introduce to you some of our work on the modeling of the mechanical tumor microenvironment and focus from the extracellular perspective on application of physical stimuli to the cells, and then to the intracellular perspective as last. So first things first, for the chemical modulation. Here on focus is our work on intestinal cells. Why we decided to focus on intestinal cells in the first place? Well, actually, intestinal cells are exposed to complex chemical mixtures. You have, for instance, in the intestine, a wide variety of food constituents, but also natural contaminants. Think about, for instance, mycotoxins, phycotoxins, argon toxins, and also chemical residues. For instance, pesticides, think about food contact materials, but also micro and nanosized structure. One examples could be microplastics. But also you have a wide variety of physical cues to which the intestinal cells are actually exposed. In the intestine, for instance, we have a very high epithelial turnover. We have the constant peristaltic movement, allowing the transition of the intestinal content along the way. You have luminal shear stress, and also at the macroscopic level, the movement of the organ in the abdominal cavity. So for the first example today, I would like to focus on natural contaminants. For those of you that maybe know us a little bit better, this might not be a surprise because we have a very strong research focus on natural contaminants at the University of Vienna. And among these, specifically on mycotoxins, thanks to the great expertise of Professor Doris Marco in the study of Alternaria alternata and their mycotoxins. So as you can see here, one of the molecules I would like to focus on today is the altotoxin 2. Among the perilenchinone structure produced by Alternaria fungi, the altotoxin 2 is actually known as quite reactive. because it carries this epoxide group that is here highlighted in violet. And it also triggers, for instance, a very strong oxidative stress, but also lipid peroxidation, just to name. So when we started wondering what is the effects of aldotoxin 2 on the intestinal cell, we actually really started from the cell surface. And one of the first things we had to look at was actually the effects of the toxin on the cell membranes. What we did actually was to measure the membrane fluidity. So we started really to see how the membrane biophysical properties can actually adapt to the presence of the toxin. We compared the two different cell lines for this project, the colorectal adenocarcinoma cells, HT29, and the human colon epithelial cells, ACC1CT. And as you can see here in this graph, for the human colon epithelial cells, we have a concentration-dependent decrease of the membrane fluidity, which means when the cells were exposed to aldotoxin 2, their membrane became progressively more rigid. Afterwards, we had a look on the membrane morphology. And what was immediately clear, having a look at the cell morphology, is actually the membranes of the two cell lines was actually quite different in the first place, where the HT29 actually have a very thick lipid-rich membrane, rich in cholesterol, and the ACC really have this very thin and filigrane structure. When we applied the toxins to our cells, what we saw was actually morphological rearrangement of the lipid on the surface of the ACC cells, but actually not much more than that. Because when we looked at the cytoskeleton underneath, we actually couldn't appreciate any difference. So it really looked that the effect of the toxin was actually stopping at the surface. Still, I have to say that this incubation actually took place in a standard microscopy slide in static condition. When we repeated the experiment in a microfluidic system, and actually we incubated the toxin in a more dynamic system, if you wish, actually the result we obtained was very, very different. As you can see here, we do have that the membrane morphology was changed substantially, and this also drugged the cytoskeleton underneath, and we can see really a very severe loss of the cell shape. So in this case, whenever we had the combination of the shear stress together with the toxin, we kind of had way stronger effect, especially on our ACC cells. This was also measurable as adaptation of the cell area spread, because as you can see here, actually the cells that were exposed to shear stress actually naturally became a little bit smaller in control condition. But when they were exposed to shear stress in presence of the toxin, they actually were kind of folding on themselves, so becoming really way smaller than the control conditions. So here we can see that actually the effects on the cell deformation were measured. So you might say these are just effects on the cell morphology. And the question is, how can we make then the further step, moving from the cytoskeleton down to the cell nuclei? In fact, the involvement of the cytoskeleton at this point is very important in relation to cell deformation, not just for the cytoskeleton itself, but also for the proteins that are connected to it. And in particular here, I'm thinking to one of our most studied mechanosensitive transcription factor, which is the NRF2. NRF2 is sensitive to cell deformation, but also to oxidative stress, because it's linked to actin thanks to the scaffolding protein KEAP1. And when it's activated, it's actually able to bind in the DNA to the antioxidant responsive elements and with this actually regulates gene transcription to obtain, for instance, phase 2 genes or elements of the antioxidant response. And here we knew already that actually the NRF2 was related to the activity of aldotoxin 2 because it was mobilized in response to application of the toxin. So what we did is actually we repeated the experiment and we looked for the NRF2. And as you can see here, we actually had quite interesting results. Firstly, I would like you to focus on the response profiles of the cells without the toxin. What we could actually observe in this case is that when we applied the shear stress at the cells in the microfluidic system, the HT29 actually did not respond in terms of nuclear increase of NR2. The signal remained, let's say, quite stable. On the contrary, the ACC cell actually showed a significant increase. We repeated the experiment in presence of the toxin. What we actually could see was a little bit different response. The HT29 actually were able to respond to the two, if you wish, triggers together, the chemical and the physical one. And in this case, we could actually measure a nuclear increase of the transcription factor NRF2, whereas the cells ACC, in this case, we lost completely the response, which is in line actually with the severe morphology loss that we could observe in the microscopy slides. So as you can see here, there is a difference in the response profile in the activation of the NRF2 if we are incubating the cells with the toxins or in the microfluidic alone combination. So starting from this very first study, it really looked like that the effects we could observe actually started from the membrane. But actually, cell membrane is way more than simply the target of ATX2. This brought us really to rethink or to study better the central role of the lipids in these processes. And this is where the second study actually started. We stayed on the intestinal cells, but we tried to elaborate more on this essential food constituent. Also knowing that the lipid metabolism is absolutely essential in supporting the growth, the proliferation, the motility of cancer cells. We saw it also in our ovarian cancer cell models that within 1 hour exposure to shear stress, they actually activate, for instance, cholesterol metabolism. So lipid metabolism is tightly linked to cancer progression. So for this study, we decided to focus on we decided to focus to dietary fatty acid. And these are actually among the most prevalent fatty acid in our diet. And these are the palmitic acid and the oleic acid. So for the sake of time, I would focus today just on the data that we collected on the palmitic acid. So when we started our experimental layout, we actually decided to change it a little bit. And instead of using the HT29 for this study, we decided to use another intestinal cancer cell line. And these are the ACT116. This is because we actually saw that the membrane of the HT29 was so different in comparison to the one of the ACC. And so we wanted to start with the cancer cell line whose membrane structure is a little bit more similar to the one of the non-transformed cells. Anyway, also in this case, we could see that the cancer cell actually took up way more palmitic acid when they were exposed to it in comparison to the non-transformed cells. You can see it here in the microscopy pictures in light blue. That actually, the ACT116, they are growing way more, especially when incubated in zero serum. And the ACT116 simply seems to have a very limited capacity. And this is actually didn't surprise us much because it's very much in line to what we saw in literature. We also measured the memory fluidity. And also in this case, actually, palmitic acid was quite active on both cell lines, even at no cytotoxic concentration. But still, also in this case, the cancer cells seem to be a little bit more sensitive in comparison to the non-transformed one. These extended also to typical mechanosensitive structures that are harbored on the cell membrane. So we had a look at mechanogated ion channels, so Piezo1, and also in this case, we could see an increase of expression when we incubated the cells ACT116 with 100 micromolar palmitic acid. So all this signaling that was described and all this effect I was describing up until now are all related to the cell surface. So what we did afterwards is actually trying to understand if this signature is actually functional. So in this case, we changed a little bit our approach and we decided to go for untargeted analysis. So we performed proteomics, comparing the response profile of treated and untreated cells in static and under shear stress conditions. And here you can see the regulated protein that we could measure in ACT116 cells after treatment with palmitic acid, comparing static and shear stress. So the presence of the palmitic acid actually changed a little bit the regulated protein profile in comparison to the control cells. And what was particularly interesting for us is that actually the cells that were exposed to palmitic acid and shear stress actually upregulated actin, which brought us one step further down in the intracellular compartment in our analysis, if you will. So we made some additional experiments. This time we tried to confirm our finding with some targeted analysis of the actin. And what we could actually observe is that actually already after 3 hours of incubation, the ACT116 started to increase the signal of the actin, a response that actually consolidated after 24 hours of exposure, and thus confirming the data that we collected in the proteome signature. We also had a look at transcription factors. Unfortunately, in this case, we couldn't perform the experiments in static and shear stress condition, but we had a look at the very, very famous mechanosensitive transcription factor, one of the most studied. And this is actually YAP1. And as you can see here, highlighted in yellow, also in this case, we have a significant variation in the cells that were exposed to palmitic acid, whereas for the CEC cells, we had no response. So all in all, the cancer cells were more responsive to palmitic acid, starting from the uptake, so from the cell surface, up to the cytoskeletal adjustment to the nuclear compartment. Still, what we saw up until now is focus on the treatment. So what I would like to do with you in the next half of the seminar is actually to move a little bit the attention away from the treatment and talk about potential model of these processes. In fact, I would like to show you some of the work we have done with respect to mechanical tumor microenvironment. Why is this important? Because actually changes in cell morphology are not just related to carcinogenesis, they're also related to what we bring in our complex cell culture model. Think about the cultivation of cells in 3D structures within artificial constraints and scaffolds. Think about the cell deformation, for instance, if you have invasion through 3D matrix, if you have deformation of the extracellular matrix, cell strain. Think about cultivation of the cells, diverse scaffolds, and the application of shear stress, as you might have, for instance, in microfluidic devices and organ-on-chip systems. So for complex cell culture models now are becoming really rich in the spotlight, especially when it comes to NAMS development. Because phasing out animal experimentation is actually bringing more and more the focus toward in vitro cell culture systems. So as far as our work is concerned in this respect, actually one of our main interests and research pillars, if you wish, in the lab is actually the study of the bladder cells. Bladder cells are quite similar, if you wish, to an intestinal compartment, because also in this case, they are confronted with a very high chemical repertoire. I'm thinking about all the metabolites that are landing in the urine, excretion products, but also they have to display a very high mechanical plasticity. So the study of the bladder cell, especially for urinary recurrent toxicant, is for us a central topic. So now I would like to talk about the work we did on the bladder cells, and especially trying to understand how they react to the mechanical stimuli. For this reason, this is one of the first experiments that we did. As you can see here, when bladder cells are cultivated with the flow, they actually, if they're having the right extracellular matrix, like for instance, they are cultivated in collagen, they're actually beautifully adjusting the cell shape. So they're becoming bigger and naturally stretching, even without any other chemical being present, any other treatment. So when we saw that the bladder cells were adapting so well to the shear stress and fluid shear stress, we thought, okay, how can we use this and make it, bring it like a step further in the study of urinary recurrent toxicant? For this, we started with another mycotoxin metabolite, and this is the DON 3 sulfate, so urinary recurring metabolite of the trichoticine mycotoxin deoxynivalenol. And a couple of years before this experiment started, this compound was actually detected in human urine. So it was first very interesting to have a look how bladder cells actually react to this compound. So we repeated the experiment, incubating our T24 bladder cancer cell in presence of shear stress plus minus the DON 3 sulfate. And as you can see in the graphs in blue, even if we didn't have any variation of the cell densities of the cells, were coping quite well with the mechanical stimulation. What we observed when applying the DON 3 sulfate is that actually the cells were spreading much more in comparison to controls. This was actually quite interesting response that we could observe there. And this probably not much important for the response itself, but actually because it motivated us to ask ourselves, why is this? What actually supports the plasticity of bladder cells, of bladder cancer cells? And since the cells have to cope with a very challenging chemical and physical environment, we started to think about autophagy as one of the potential players. As you can see here in the images, these are the experiments that we repeated with the microfluidic system in presence of a very widely used autophagy activator, which is rapamycin. So we incubated the cells with different concentrations from 1, 10 to 100 nanomolar, and actually rapamycin returned a very similar response in comparison to DON, so an increase in cell spread. This we also quantified because it's quite a little bit difficult to appreciate it from the bright field images. And as you can see here, we have a concentration-dependent effect where the cells were getting progressively bigger and spreading more, adhering progressively more to their substrate when they were confronted with both the rapamycin and the shear stress. So we thought this could actually be molecular mechanism also for our cells. And what we actually did here is we repeated the experiment comparing studied incubation with shear stress incubation. As you can see here, we have the DON3 sulfate in blue, the DON3 sulfate plus chloroquine in green, and in yellow, the chloroquine. Chloroquine is a commonly used autophagy inhibitor. And if we incubate the T24 bladder cells in static condition, we actually have no variation of the cell area. If you look at what happens when we have the shear stress incubation, what we could actually measure is that the cell area spread triggered by the deoxynivalenol-3 sulfate when actually blocked by the presence of the autophagy inhibitor. So actually here we could mechanistically support that probably autophagy might play a role in the mechanical adaptation of bladder cells to the shear stress in combination with the toxin metabolite. So we realized that actually the effect on the adaptation of the cell morphology was not everything we could measure. And particularly in respect of adaptation of cell morphology, we started to think if maybe we could have an effect stemming from cell adhesion on the capacity of the bladder cells to move, particularly in the migration. So in a follow-up study, we decided to try to move a little bit from the cell surface toward movements that also affect the intercellular compartment. So we repeated experiments of migration assay, and we compared the motility actually of the T24 cells in presence of absence of rapamycin. So as you can see here in the box plot that are in orange in full color, when we applied the rapamycin, what we actually obtained is a reduction of the cell migration. This is compatible with the notion that application of the autophagy activator could actually increase the cell spread, probably cell adhesion to the surface, and possibly slowing down motility. We also repeated the experiments in presence of shear stress. And what we could actually see here was a further decrease of the cell movement, also compatible to the response we obtained with the microfluidic system. So we thought maybe similar molecular mechanisms might be involved here, like we previously observed for the DON. So we repeated the experiments in presence of autophagy inhibitor. This time we use bafilomycin. And we also combined bafilomycin together with rapamycin, following a similar layout as we previously described. To our disappointment at that time, actually, we realized that also when we incubated the cells with the autophagy inhibitor and the combination of the autophagy activator and inhibitor, we actually obtained always a reduction of the cell motility, which in the end brought us to the point that other mechanisms of toxicity might have been involved in what we were observing. So we started again from the cell surface, and we asked ourselves if maybe the rapamycin can affect the cell membrane. This time, we actually could see no effect. There was no changes of the cell morphology, and a little bit fluctuations in the membrane fluidity maybe, but not really substantial effects on the cell membrane. Then we moved down in the intracellular compartment, starting from the surface, like caveolin 1 as a cell mechanosensor. We couldn't see any variation of the signal. Intensity of actin cytoskeleton was actually decreasing. Still, what we saw was a rearrangement of the integrin signal, and also intracellular rearrangement and adaptation of the actin cytoskeleton within the cells, measured as actin area per cell. The application of rapamycin actually returned a reorganization of the cytoskeleton. That was not the only thing that we observed. We also saw a decrease of the mitochondria, both in signal and the surface, and very interestingly, a decrease of the intercellular acetylation. And here is the graphs of the cytoplasmic region. So we went back to literature, and what we actually saw is that there are NADA plus dependent deacetylases called sirtuins that actually are involved in mitophagy, which would be plausible with us incubating our cells with autophagy activator rapamycin, but also that the acetylation is absolutely crucial in regulating cytoskeletal dynamics. So what we did was repeating the experiment with the sirtuin-1 inhibitor nicotinamide. And as we were hoping for, we actually could see that the application of the nicotinamide significantly increased the acetylation in the cytoplasmic region of the cells. It also reverted this acetylation decrease that rapamycin triggered. But what was most important for us, it not just recovered the acetylation, it also restored cellular motility. So as you can see in the graphs in orange, the application and co-incubation of the nicotinamide together with the rapamycin actually restored the motility of the cells in the migration assays to a level similar to that of controls. So this finally brought us to the point that there is a lot going on in the intracellular compartment. We identified clearly the central role of autophagy, but still there was the open question of the response to bafilomycin. And this somehow brought us our attention to the intracellular compartment, and particularly to the interaction and the rearrangement of this intracellular compartment with respect to cell deformation up to the nuclear region. And this is the last work I would like to present you. We published this year, really trying to tackle molecular mechanisms that affect the intracellular compartment and its deformation towards the nuclear region. For these experiments, we actually started with bafilomycin. And we started from the observation that even short-time incubations, we are talking here about a couple of hours, 4 hours, triggered a concentration-dependent rearrangement of the mitochondrial network. This we could actually measure as a decrease of the mitochondrial area signal. And as you can see, the spread of the mitochondrial network becomes progressively smaller with the organelle clustering around the nuclei. This was actually not accompanied with oxidative stress as far as we could measure. And what we also wanted to observe is actually what happens in the cells when we have this intracellular rearrangement. For this, we started to think about measuring the biomechanical properties of the different intracellular compartments. We did, as we previously optimized, some atomic force microscopy experiments that actually allows us to measure the stiffness, the Young modulus, in different subcellular compartments and distinguish between the nuclear region, the perinuclear region, and the cytoplasmic region. With this technique, we could actually appreciate that the intubation with baphophilomycin actually triggered changes in the intracellular stiffness. And what was particularly important for us was the observation that actually we had significant increase of intracellular stiffness related to the nuclear and the perinuclear area, exactly where the mitochondria were clustering. So we repeated the experiment in presence of an antioxidant catalase to have a little bit of an idea on how these responses can be eventually affected by the presence of the antioxidant. And as you can see here in the figure, actually the movement of the mitochondria triggered by baphophilomycin was relatively constant, even in presence of catalase. What was also for us very, very important is that this mitochondrial rearrangement actually occurred when the cell area stayed constant. So it was not an artifact as far as we could measure from the cell's holding, for instance, in itself. And also the intensity of the actin cytoskeleton actually was maintained stable. So we repeat the experiment of measuring the nuclear stiffness. And also in this case, we had, like for the concentration-dependent effect, quite robust increase of the nuclear stiffness when the cells were incubated with baphophilomycin or with baphophilomycin plus catalase. Importantly, this was also associated with a significant increase of gamma-H2AX signal. So that made us suppose that this nuclear deformation and increase of the nuclear stiffness that we could measure could actually be associated with a genotoxic damage. And this would be actually in agreement with literature, where it was recently reported that cells that undergoes a very tight constrained migration, like for instance for metastasis during intra or extravasation, this makes the nuclear deformation that is necessary and that enables this process actually the cell prone to genetic instability. We also performed some further experiments to measure the genotoxic damage. So we measured COMET assay, and also in this case, we could observe a very consistent response. So all in all, our current working hypothesis, and that we're trying further to deepen with experimental work, is that actually the nuclear deformation that is actually related to this intracellular rearrangement could actually support the increase in DNA damage that we might see in the cancer cells. So it's now time for me for some conclusions. Starting from the fact that there are molecular cascades related to cell deformation, and I hope I could show you that this accompany both molecular mechanisms that are essential for carcinogenesis, but also when we apply complex cell culture models. Related to this, that chemical and physical cues can leverage on the same intracellular pathways. We saw it, for instance, with the NRF2, where we can have chemical-driven activation, but also physical-driven activation. And this has a great relevance and great potential, in my opinion, if we are trying to produce models that are always more refined and actually representative of the human physiology, and that can be used also as an advanced in vitro model. And also, why not, to eventually identify some experimental confounding factor. And with this, I am at the end of my talk. I really would like to start here with a big thank you. And the first person I would like to thank is actually Professor Doris Marco, because I don't know if you have noticed, much of the work that I've presented today started when I was a postdoc in her team. And she was the one that enabled me to follow these stories that I'm now have the pleasure to continue with my own team. For the data I've presented today, Janice Bergen and Maximilian Jobst, they are my experts, if you wish, on intestinal cell models and bladder cell models, but also the whole biophysical toxicology team and the departments, food chemistry and toxicology, analytical chemistry, and obviously the funding support. I thank you very much for your attention, and I'm happy to take your questions.
Thank you very much, Giorgia. Excellent promotion for the next EUROTOX meeting in Vienna with your picture. Can you hear me? Yes.
Yes, we were at the booth in Athens.
Yeah, and
This is
I
just
must say
one of
your
the
presentation.
many adventures Doris and me had together.
I must say your presentation was very clear, very comprehensive, and very, very interesting. Really discover some new things. You had some. I have one question in the chat and a great compliment from William Bisson that had to leave because it's Thanksgiving in US right now. We have one question from Amaya Irizar. So thank you for an excellent presentation. Would you expect similar behavior to the surface exposure of chemicals with lower track respiratory epithelial cells? What would be the main challenge to study this mechanism in respiratory culture model?
Well, I have to admit that I have never had the pleasure to work with models of the respiratory tract. But I know that lung tissue is also on focus, especially with respect of development of organ on chip technologies, because also in this case, we have highly motile tissues and contraction that physiological plays a role. So I would expect that also in this case, we might have a crossroad of different pathways going on. And obviously, I think the challenges are very much dependent on the cell types, at least to our experience, to the cell types you're starting with. This was, for instance, the case. We realized that the bladder cells, for instance, have a very high autophagy competence in the first place, because this is very necessary for them for their physiological function. And this is where we build on. So I guess that this will be much tailored on the model.
I have a question related to the beginning of your presentation, so concerning the membrane fluidity modification. So at the end of the day, you have, if you don't put shear stress, you have very few effect on the actin skeleton. So finally, at the end, what do you think is the mechanism of membrane rigidity induced by the toxin? Because there is also other chemical that can provoke membrane modification, like nanoparticles. So what do you think, where the rigidity is coming from? It is modification of lipids or?
So we didn't have the capacity to test it for this project, but we explored this with the ultratoxin 2. And we could actually see the application of altotoxin 2, for instance, in the THP1 cells, so immune cells that triggers changes in lipid peroxidation. I guess what we could see here related to the changes in membrane fluidity is related most likely to lipid peroxidation. What I didn't have the capacity to show you is that we also did matching experiment with N-acetylcysteine, and there we could see recovery of the cell membrane fluidity as hampered by the ATX2. So I very much assumed that this was related to oxidative stress mechanism. And then obviously, when we applied the shear stress, we kind of forced the response of the membrane. But we also had a very similar response when we measured the migration, because obviously cell has to adapt with the membranes in the migratory process. So we had matching, let's say, data with the migration, and that brought us a little bit to the point that the attack on the membrane might be of limited, let's say, importance or severity when the cell is static. But whenever the cells try to move or is exposed to movement, then this becomes more critical.
Okay. I have a general question from John Anetor. Could you please comment on the implication of this mechanism, I guess, on protection against malignancy of the gut, if it's possible?
Well,
I could see food.
Yes, I think for me, when we first measured this shear stress dependent activation of NRF2 in the intestinal cells, was a little bit of an epiphany, if you wish, because when I first studied and when I tried to understand what is the effect of the differential exposure of intestinal cells to different dietary constituents and contaminants, I thought that maybe the speed of the transit might regulate primarily the exposure. So if it goes slower, might be we have a differential exposure than if it goes faster. But then I realized that there is the second part of the mechanism related to the potential of the intestinal cells to actually activate antioxidant responsive elements in response to the flow. It's probably not a surprise that when you have cells and intestinal cells cultivated in microfluidics, they tend to differentiate faster and more efficient and in a more structured fashion. And that probably relates to the fact that the intestinal cells actually needs these mechanical cues in order to function, if you wish, at their best. I think that this mechanisms also brings about the differential response of the different cell lines to the eventual food constituents and contaminants. But obviously, to bring it to the effects on development of intestinal tumors is a little bit of way. It's a way to go. Our studies are very mechanistic.
Okay, thank you. So I have also a question on the last part, because you're showing that nuclear deformation is associated with genotoxic damage. So what did you look at? What did you look at? Mutation, oxidative mutation due by oxidative stress on DNA, or is it also applying to chromosomal damage like carcinogenicity, for example? Or it's only you look only at real genotoxic mutation?
Yes, unfortunately, we also didn't manage experimentally to answer this question yet, because this paper was published just this year. So this is exactly the point where we are. We are developing the models in different directions, but this is definitely a question that will have to be answered. Also with respect of the potential reversibility of the effect that we observed.
Okay, so because there's no more question in the chat, but me, I have some questions, so I can use your time until 2 o'clock. So, because it's, I mean, it's very, very interesting. It's very, very interesting. So I have a very, very general question because I guess some other people can think about the same question. I mean, how can you use your data and your models, because you talk also about NAMS model, and these 8 points to in some way integrate them in predictive toxicology. I mean, for genotoxicity or maybe carcinogenesis, or where can we use this endpoint, and what kind of model we could use, because you suggest that we could use some NAMS approach.
Mm-hmm. I think that these pathways have great potential when we think about adverse outcome pathways, because we have different molecular initiating events and different key events up to the adverse outcome. So I think that there is much possibility to potentially integrate these steps in the cascade. This is probably where I could see your first application, because we try to actually describe molecular mechanisms. And I have to admit, when we started thinking about papillomycin, we were not envisioning that this could be direction on the genotoxic damage. This is not something that you would typically observe. We have, I think in this study, we were at the beginning, our results are quite tightly related to the model, because what I didn't manage to show you is that we repeated these experiments also with different stem lines. And for instance, for the ACT 116, we couldn't appreciate any movement at all. We couldn't appreciate any nuclear deformation, probably because these cells are quite small and tightly packed. We also repeat the experiments with the OFCAR cells, with the SCOF3 cells, ovarian cancer. And in that case, we saw that we could see a similar phenotype, but starting from higher concentration. And then we went back to our data and we realized that actually the SCOF cells are way more rigid in the first place in comparison to the bladder cells. So I think what we see here is a little bit of a combination of the cell type being so compliant, if you wish, from the mechanical point of view, and the chemical application. So I think it's a little bit of a game to try to combine the two effects together.
Okay, so I have no more questions. There's no more question in the chat. So I will go back and read your paper in eScience, that's for sure.
Thank you. It's always a pleasure to hear this from the colleagues.
Yeah, really. I mean, it's really passionate. I'm really interested to see the relationship between the mitochondria and the nucleus deformation. So again, thank you very much, Giorgia. I think it was the presentation was very well appreciated by all the colleagues. And I wish you a very nice day, and a very nice day to all the colleagues who attend the webinar. Thank you very much. Bye-bye.
Thank you.
Thank you. Bye-bye.
Bye.